And the next presentation will be given by Dr. Li Kong, Effects of matrix metalloproteinase 7 on vascular functions in mice with atherosclerosis. Dr. Kong, please.
Okay, good morning, everyone. I'm Li Kong from the University of Hong Kong. Currently, I'm PhD student now. So today, I'm honored to be given the opportunity to present my work here on the relationship between the matrix metalloproteinase 7, MMP7, and the vascular function in mice with atherosclerosis. So as we know, atherosclerosis is underlying the cause of cardiovascular disease, including the coronary artery disease, CAD, and stroke. So which is a chronic inflammatory disease characterized by the buildup of the lipids in the lumen of the arteries. The pathogenesis of atherosclerosis involves the buildup of the lipids, the foam cells, and when resulting the atherosclerotic rupture, so the formation of the blood clots lead to the blockage of the blood flow. To date, many clinical studies have demonstrated that there is association between the MMP7 expression and atherosclerosis. So for example, the plasma levels of the MMP7 in patients with carotid atherosclerosis showing the ischemic symptom. And also, the MMP7 levels is detected in the macrophage-rich and collagen pore area of the human carotid atherosclerosis plaque, but not in the healthy human arteries. So as the smallest member of MMP7, the MMP has the proteolytic enzyme that cleave the matrix components such as the fibronectin, the gelatin, the collagen, elastin, and laminin. Besides the macrophage, the microarray experiments demonstrate that the gene expression of the MMP7 is highly upregulated in regenerated endothelial cell of the porcine coronary artery after balloon injury. So it is well established that the atherosclerosis is associated with the endothelial dysfunction. So although some studies demonstrate that there is association between the MMP7 levels and atherosclerosis, the effects of MMP7 on endothelial function has not been studied. So our endothelial cell play an important role in regulating the endothelial function by releasing some signals such as the nitric oxide and the prostanoids, so which are synthesized by the nitric oxide synthase and cytooxinase, respectively. So our endothelial cell regulates the relaxation and contraction of the smooth muscle cells. So now in the present studies, the effects of MMP7 on the endothelial function has been examined. So some experiments were now performed in the male white-tail mice, MMP7 null mice, ApoE null mice, ApoE MMP7 double null mice, fed with 15 weeks half-a-day from the age and die weeks 5 weeks onwards. So this is a plasma of these mice will be collected for measuring the lipid levels, and the alta were dissecated for assessing the degree of the atherosclerotic plaque with the Royal Old O staining, and the vascular response with the Weyermeyer graph, and the protein levels with the Western immunoblotting. So after 15 weeks half-a-diet, the body weight in the MMP7 knockout and the ApoE knockout mice is lower compared to the white-tail mice group, but the decrease was not observed in the ApoE MMP7 double null mice. On the other hand, the systolic blood pressure in MMP7 null mice and all the ApoE deficient mice was comparable among the age and diet matched white-tail mice group. And then the plasma was collected for measuring the triglycerides, the total cholesterol, the low-density lipoprotein, the high-density lipoprotein cholesterol. So the plasma levels of the TG, TC, LDL in MMP7 null mice are not different with the white-tail mice group, but was significantly higher in ApoE null mice and ApoE MMP7 double null mice. And in HDL levels, the plasma level of the ApoE knockout and ApoE MMP7 double knot mice was higher than the wild-type mice. So this is a result as such indicated that the MMP7 deficiency does not affect the lipid metabolism on the ApoE knockout mice. And now, as showing as the oil-red outstanding, as we can say, the atherosclerotic plaque was present in the ApoE knot mice and ApoE MMP7 double knot mice. But the degree of the ApoE atherosclerotic plaque was smaller in mice without MMP7. And then to examine the vascular response to vasodilator, so the aorta was first contracted with phenylephrine, and then relaxing with the concentration increasing to the acetylcholine. So acetylcholine, which is the endothelin-dependent relaxing agent, and the acetylcholine caused the endothelin-dependent relaxation was with the endothelin, and the effects were not different among the aorta for the different mice group. And then the acetylcholine, the reaction to the acetylcholine were abolished by the LM. So LM, which is the nitric oxide synthesis inhibitor. So this is a result as such indicated that they were mediated by the nitric oxide. And then in the presence of the cytooxinase with indomethacin did not have any significant effects in the endothelin-dependent realization curve of acetylcholine in phenylephrine contracted aorta. And then in the presence of the indomethacin, so LM also abolished the acetylcholine-induced realization in the foregroup of these mice. And in aorta without endothelin, so contracted with phenylephrine, so the NO donor, which is nitric oxide donor, ranging from 10 to minus 9 to 10 to minus 4 molar, induced the concentration-dependent realization. And the endothelin-independent realization were comparable among the wild-type mice, MMP7 null mice, ApoE null mice, and ApoE MMP7 double null mice. So this is a result indicated that the MMP7 deficiency does not affect the smooth muscle response to the nitric oxide in the mice with ApoE with or without ApoE. And then in aorta, in quiescent aorta with endothelin, so the acetylcholine, when the nitric oxide synthesis is inhibited by the LN, so the acetylcholine caused the endothelin-dependent contraction only in ApoE null mice, but not in the wild-type mice, MMP7 null mice, ApoE MMP7 double null mice. So based on the previous studies in the literature and in our laboratory, so that is the endothelin-dependent contraction to acetylcholine and in the ApoE mouse aorta. And this is the endothelin-dependent contraction was mediated by the cytooxinase-derived prostanoids and by activation the thromboxane prostanoids receptor. Although the MMP7 deficiency prevents the development of the endothelin-dependent contraction in ApoE null mice, it does not affect the sensitivity of the thromboxane prostanoids agonist prostanoids in the smooth muscle cell without endothelin. So this is a contraction to the thromboxane prostanoids agonist U46619 induced contraction were comparable among the foregroup of these mice. Therefore, we would like to examine whether or not MMP7 deficiency does not affect the generation of the endothelin-dependent contracting factor in ApoE mouse aorta. So with the Western immunoblotting, the protein levels response to the enzyme in the endothelin-dependent contraction isoform, the COX-1 and the COX-2 were examined. So while the protein levels of the COX-1 did not have any significant effects among the aorta of the different mice, but the protein levels of the COX-2 was increased in ApoE null mice group, but the upregulation were not observed in the ApoE MMP7 double null mice. So the change to the COX-2 protein levels in the ApoE null mice and ApoE MMP7 double null mice appear to correlated with the MMP7 protein levels. So in summary, our research has demonstrated that the MMP7 deficiency attenuates the development of the atherosclerotic plaque without affecting the plasma lipid file in the ApoE deficient mice fed with half a diet for 15 weeks. And in addition, the MMP7 is upregulated in the ApoE deficient mice fed with heifer diet, and the upregulation of MMP7 is associated with the upregulation of the COX-2. So in the ApoE deficient mice fed with 15 weeks heifer diet, so which in turn account for the endothelin-dependent contraction to the acetylcholine. And as a result, this is a finding suggests that the inhibition of the MMP7 induction in mice with hyperlipidemia prevents the endothelin dysfunction, and hence the progression of the atherosclerosis. Last but not least, I would like to extend my heartfelt to my research team member for their assistance and the financial support of the University of Hong Kong Research Committee for this study. So thank you, audience, for your kind attention.
Thank you very much, Dr. Kong.
Thank you.
The paper is open for discussion. Are there any questions from the audience? Yes, please.
Hi, Chen Xue, Helena Qing from Monash University. Thank you so much for the excellent presentation and very interesting data package. I was wondering, have you looked at the levels of nitric oxide in the blood vessel and the expression and activity of eNOS or iNOS in the different animal groups? Thank you.
Okay, thank you for your question. Actually, we used the plasma levels for measuring the nitric oxide levels, the amount of nitric oxide. And we observed that the ApoE null mice showed the lower the NO, the amount of the NO in the ApoE null mice. And also, as well as we measured the phosphoinos and total enos in the foregroup of these mice. And this is a results show that the ApoE null mice still have the lower the expression of the phosphoinos. And the ApoE MMP7 double null mice was higher the phosphoinos in the ApoE null mice in the phosphoinos protein levels. Thank you.
Great, thank you. Thank you so much.
Thank you. Welcome.
Thank you. Number 2, please.
So what do you think is the mechanism, underlying mechanism that's regulating the decreased expression of COX-2 by MMP7? Well, since it is like extracellular matrix metalloprotease, so it is, my impression is the function of this kind of enzyme is regulating the function in the extracellular milieu. So why it would influence the intracellular expression of COX-2 and even the function of the vasculature?
Thank you for your question. That's a good point, actually. From the increasing the report view that the endothelin-dependent contraction EDCF to the acetylcholine is activated by the phospholipids, the PRA2. So among the phospholipids, the PRA2, the cytosolic CPRA2 will convert the phospholipids to activate the COX cytooxygenase, the prostanoid receptor, and then once released to the cytooxygenase, so it will be converted into the arachidonic acid to activate the cytooxygenase, the prostaglandin, the peroxidase. So once it releases cytooxygen-derived prostanoids will activate the surboxane prostanoids and increase the vascular function to contraction. So this is the mechanism for the endothelin-dependent contraction.
Thank you. You presented here that in MMP7 knockout animals, they had some benefit in your models. So do you have any pharmacological means that you would like to try to result in reduced MMP7 in your animals?
Yeah, we just use the pharmacoinhibitor of the MMP7, like the GM981 and doxycycline, for the explore the effects of the MMP7 enhanced endothelin-dependent contraction. And the present studies show that it will, after incubated with the GM981 and the doxycycline, will be inhibited the endothelin-dependent contraction in ApoE null mice. Yeah, thank you.
And thank you very much, Dr. Kong.
Thank you.
We have two other presentations in the list, Dr. Xu and Dr. Xia, but my understanding is that they have not delivered the presentation, but are you present here in the auditorium? If no, thank you. Thank you to the two speakers here, and thank you everyone for coming to this early morning session, which we now close, and the next session here will start at 9. So thank you and have a nice day here in Glasgow.